So today I'm going to be speaking more from the microbiology perspective and the bacteria in general, not so much about the specifics of this product and what it does, but just the bacteria in the soil and its relationship with plants. So I'm going to take you back first off on a way back history. Go back to that slide. We understand agriculture to be about 10,000 years old, and that as humans, we needed a better stable supply of our food. So we decided to get control of plants and make crops and earn our food that way. However, the ability to do this actually started much earlier, almost 4 billion years ago, with the solidification of the earth. So soon after the earth cooled to a solid state and we had crust, you had the formation of soil, which I'll call ancient soil. This is a very primitive soil, which is basically dissolved minerals that were running off the crust or being eroded from the crust with the newly formed liquid water. Soon after this, you have the establishment of microbial life on this ancient soil. So the earliest microbes were able to live and thrive in this dissolved mineral crust that we called ancient soil. That persisted for about 300 billion years as a standalone environment for the new life that was being formed on the planet. And it really started to move in leaps and bounds to something that we consider more useful today about 700 million years ago with the establishment of lichens. So a lichen is an interesting species. It's the formation or the organization of these single-celled organisms. So the bacteria, the algae, and fungi came together and worked together to make a thriving organism, which is called a lichen. We still have them today. They are old. They permeate some of the most austere conditions on the planet. So they're able to live under very harsh conditions. And they were the first step, or the not so missing link, between the fungi and the other single-celled organisms and what we now know as multicellular plant life. So the lichens, really what happened is they were able to take the life and move it away from the water's edge more inland. From this, we had the establishment or the evolution of novel plants. And the earliest plants didn't even have a root system. So they still used the lichens and the fungal systems that were existing to act as their root system. Soil as we know it really only developed after plants about 380 million years ago. So as we had larger plants and more sophisticated plants develop and recycle through the process of life and die, we started to add organic matter to this ancient soil, which made it much more akin to what we're used to today. I don't think I have control anymore. Okay, there we go. So the soil that we have, or the soil in general, is alive. And it's important to remember this, that the dirt that is ubiquitous and you see on the ground is actually teeming with life. So in a single gram of soil, you can expect to find billions, if not tens of billions of microbes living and thriving in their own little world. Typically, there's 5 that contribute to this biome, this population: bacteria, which is the focus of this particular conversation; fungi, which are also very important and almost as numerous as the bacteria, but not as quite. And to a lesser extent, you have algae, which we spoke of earlier, and protozoans. Protozoans are actually single-celled animals in the most simplest definition, as well as viruses. And as much as we'd like to think that viruses are the bane of our existence today, given this pandemic, they're actually very important in the soil microbiome, and they're very under-researched. So novel research is really starting to focus on their role in the microbiome. But as I mentioned at the top of this, we're going to start with bacteria, or we're going to speak about bacteria for today's talk. Besides the microbiome, which I've mentioned here, there's also worms and insects, which are multicellular, which are things that you can see. But if we were to take all the bacteria from a population perspective that's living in that 1 gram of soil and think about it in more macroscopic terms, it would be the equivalent of having 2 to 20 cows on an acre of land. Similarly, if we were to add up all the weight of the bacteria in an acre of soil, you'd expect to find 1 to 1.5 tons of mass. So it is a significant portion of our soil, and we need to remember soil is alive. And like any living organism, it thrives and it can be healthier or less healthier given conditions. It's not moving again, Shiva. So the soil microbiome underpins the productivity of all agricultural enterprises. And at the intersection of the three ways that we would speak about soil or soil fertility is bacteria. I can't see anything now. So at the intersection of the three ways we can measure or speak about soil fertility is bacteria. And they play an integral role, and the interplay between these is really controlled by them. So for example, if I were to speak about nutrients, we know that the bacteria, that we've heard a number of times this morning, are able to cycle nutrients or produce nutrients that are useful for the plant. In doing this, they may change the pH or the salinity of the soil, but that in turn may work against the ability of the plant to uptake the nutrients. As well, if we were to speak about organic matter, which Dr. Bill had introduced to you, the degradation of this matter is done predominantly by bacteria and fungi. When they degrade or when they decompose the material, they're actually changing the organic matter or the content of the soil, which in turn changes the texture or the holding capacity of the soil. So physical, chemical, and biological fertility are really not three standalone topics, and they are controlled, or they are actually the interplay between them is really controlled by the bacteria in that soil that we spoke about earlier. Next slide. So when we talk about soil, it's important to note that not all soil is equivalent. So even though we look at a field and it seems relatively insignificant to us, there is zones within that soil that are much more important from an agricultural perspective. And one of them would be the rhizosphere. So the rhizosphere is actually a very small or narrow zone that sits in and around the root structure of the plants. It's a sophisticated transport and communication zone between the root plants as well as the surrounding environment, so all the microbes. And it actually can extend anywhere from, say, 1 to maybe upwards of 3 millimeters away from those roots. So it's a very narrow zone. However, it's a very, very busy zone. So we can consider it the head office for the soil microbiome, in that a lot of activity, the highest level of microbial activity is going to occur in this rhizosphere. Now, this is as compared to the bulk soil, which gets further away from those roots, where you still have activity, you still have bacteria and all the microbes that were mentioned before, but you don't have the level of activity. And the rhizoplane specifically is that immediate interface. So right where your soil is touching your root, or right where the microbial biome is touching the root, is considered the rhizoplane. So that's even more of a specialized zone. Next slide. So this is kind of a busy, complicated slide, and I don't want to take much more away from this, or want you to take much more away from this than what's in that top right corner there. So we spoke about the rhizosphere in the previous slide and how it's teeming with this bacterial life, and there's a lot of activity in a very narrow zone. It's important to note that the rhizosphere itself also has a gradient in the chemical, biological, and physical properties. So what you will see longitudinally or up and down a root shaft is going to be different from 1 micron to the next, depending on the type of root or the type of cells in the root zone you're in, as well as what bacteria might be there or what fungal relationships are having with the root. This also changes radially. So this would be moving away from the root, which I mentioned earlier when I said it's about a 1 to 3 millimeter zone. So if you look at the graph in the top right there, you can see that as you get further away, your microbial diversity will increase. So the number of pathogen, or the number of bacteria that you have out there, are going to be larger. However, the density of them will be less, as well as the activity or the specialty functions that they perform out there. So really the business center, as I mentioned before, and the important zone when we're speaking from a microbial perspective, is going to be in that rhizoplane and rhizosphere, which is very close to the root. Next slide. So just to come away from the root for a little bit, to give you some background on bacteria. Bacteria are some of the oldest living organisms on the planet. These are actual real life pictures of bacteria. The color is artificial, but using high-powered microscopes, this is what they look like when we intensify the magnification. They come in the 3 typical shapes, which are spheres, rods, or spirals, and usually in their name, it'll indicate the shapes that they are. Next slide. So like I mentioned earlier, they are ancient. They appeared about 3.5, give or take, billion years ago. they're a single-celled life form, so they're considered simple in that they don't have this higher order of organization. They are able to commit all of their life processes within a single cell, and they reproduce by binary fission. All this means is that they take everything they need in that cell, they double it up, so they double all their materials, and they split in half. So one cell becomes two. Next slide. Now, that's important because we're going to speak about the Darwinian goal or the purpose of life. So being the most basic life form on the planet, bacteria adhere exclusively to the most basic purpose in life, which is to produce offspring that in turn produce offspring. So the history of life is really one extended match of genetic brinkmanship, also known as evolution. As we've started from these single-celled simple organisms, the ability to increase our chances to live, to have offspring, has allowed our genomes or has forced our genomes to evolve to what we consider today with higher order primates and other sophisticated animals that we see on the planet. Next slide. So it turns out that the reason for this, or the purpose for this, is that life is a struggle. So microbes are in constant competition for resources. So all they want to do is grow and divide and grow and divide. However, it's not that easy to be successful, or certainly be successful in the numbers you need to when you're out competing millions, if not billions of neighbors. So to do this, they've evolved a number of strategies. This has come, as I mentioned before, from the evolution of their genome. And if we think about them as single-celled germs that are very simple and very primitive life form, we have to really stop and check our thoughts there, because when you look at the number of genes that a bacterial cell has relative to a human, it's actually only 10%. So humans, although we're approximately 30 trillion cells in this highly organized fashion with well-developed differentiated organ systems, including a brain, the bacteria, that much smaller and seemingly that much more simple, still only have 10% of the genome that we have. So that means that they have really taken this evolution thing seriously in developing all the tools and all the weapons they need to allow their success just to produce offspring, just to grow and divide. So some of these include structural changes like a layered cell structure, so permeation, protection from your environment, like walls around a house. You also have some weapons involved when you talk about antibiotics and endotoxins. So these are actually substances or chemicals that bacteria can exude or excrete into their environment, hopefully taking out their neighbors, reducing the competition and allowing the cell to get more access to resources. Other ones have to do with more specialized functions like quorum sensing, so the ability to send and receive signals in the form of chemicals in the surrounding environment so that you can know and understand what's out there and what I might need to do to change, as well as specialized metabolites or secondary metabolism. So the ability to process or produce alternative chemicals that aren't necessarily needed for base life processes, but are able to help the bacteria outcompete when we're talking about austere conditions. Next slide. So when we apply this thought to the rhizosphere, life is still a struggle in the rhizosphere. And the examples of the adaptations that we expected or we have come to understand bacteria have developed include spatial. So living close to your food. So when we said the rhizosphere is about a 3 millimeter maximum zone, why is that? Well, the primary reason for that is because they want an energy source. And the closer you are to your energy source, the more likely you are to have a large, healthy, fruitful life. So 10 to 40% of the photosynthesis fixed carbons in plants is actually in the root or released from the root. So this ends up being a very rich carbon source for bacteria. So this is one reason that you're going to find more bacteria near the root as opposed to further away. Also chemicals. So the bacteria produce degradation enzymes like proteases, which break down proteins, amylases, which can degrade starches, cellulases, which is cellulose, a very complex carbohydrate that plants have in its structure and its wall structure. And these are typically inedible to smaller organisms, which are really just looking for simple sugars. But through the production of particular enzymes or degradatory enzymes, the bacteria are able to systematically break down these much larger, more complex molecules into a food source. Another development is finding alternative energy sources altogether. So sometimes there's just not enough carbon, or there's just not enough usable carbon, or there's just too many people trying to source the carbon. So what the bacteria in some instances are able to do, they're able to change their metabolic pathways or even just employ completely different metabolic pathways so that they can use other sources of, for energy, such as nitrogen, maybe phosphorus. There's even some sulfur using bacteria out there as well. Next slide. So the Darwinian principle in the context of bacteria actually applies to plants as well. So the basic purpose of a plant is to produce offspring that produce offspring. So life is a struggle for them as well. To pay for this process, which is it turns out an expensive process to commit, plants have developed a sophisticated system that spans both subterranean environments as well as the above ground. So photosynthesis, which is the one we're all familiar with, where plants utilize the sun's energy or exploit the sun's energy to take carbon from the air and fix it into these more complex hydrocarbon molecules, happens above ground. And we've got the important structures in the leaves, the satellites that actually catch the sunlight, as well as your stems, which provide support. However, underground, you have an extensive system that the plant has developed to exploit the soil, where it gets the water and other nutrients that it needs for its life processes. So the root system really talks to the top and vice versa, and is able to take the best of both worlds and maximize its environment as a whole. Next slide. And as an example of this, you can see that root systems, which are the communication and transport infrastructure of the plant, are incredibly developed and oftentimes can be larger, 2 to 5 times larger than what you'll see above ground. So although we're most enamored with what's above ground and we like the way the foliage and flowers looks, and that is mostly where our yield comes from an agricultural perspective, barring, I guess, tubers and some underground vegetables, it's this root system that really allows the plant to produce and grow above ground and really is important in plant health and crop yield. Next slide. So the plant microbiome, we can consider these two really to be the original gangsters of outsourcing. So you're looking at this 380 million year old relationship that has, like any relationship, taken time to work out the kinks and develop an ideal system where each can act as a partner in it. It looks altruistic, and altruistic by narrow definition is really getting satisfaction from some sort of a selfless act. But as a biologist, I can tell you that altruism does not exist in any biological system. It's all really quid pro quos and transactions that I'll scratch your back if you scratch mine. And this is an excellent example that we see when we're looking at soil microbiome or the microbiology of the soil in relationship to the plant. So the plant roots, we talked about earlier when we were speaking about the Rhizosphere, exude or are able to release nutrients and chemicals that can act as a food source for bacteria. Sometimes these are directly usable, so the bacteria can just burn the carbon. Other times the bacteria themselves have to further process and degrade these to get the usable carbon energy from them. These processings or these metabolic processes that go on in the bacteria allow or produce molecules that are more useful to the plant. So Dr. Bill spoke extensively about the nitrogen cycle, and really what he's trying to mention was that the most usable currency of nitrogen, which is very abundant on the planet, it's probably the most abundant element out there, but the most usable currency from a biology perspective is really the form of nitrate, that NO3 negative that you see on the screen there. Other forms are usable, but they're not necessarily available, and some are just totally useless when it comes to uptake and use in a living system, in a biological system. Same thing with the phosphorus. It's an incredibly important molecule. It forms the backbone of all genomes. So DNA relies extensively on the phosphate molecules, but it also is a little bit tricky because it's needed in 2 narrow chemical moieties, which we see there called the orthophosphates, but any other form in the most common forms are really not usable by plants or by animals. So in this instance, the bacteria is able to produce these more usable forms of nutrients. The plant can take them up more readily and use them in their life processes, and they're absolutely necessary for the life processes. The addition we get is growth hormones. And growth hormones is a little bit interesting, talking about that altruism or that quid pro quo. The bacteria produce these chemicals that look or act as in growth hormones. They, in turn, promote growth of the plants. So when you have additional growth in the root zone, you have the shedding, the natural shedding and reducing of older materials on the outside. This shedding or older materials is then utilized by the bacteria as an energy source. So what actually looks like a favorable offering in the form of a growth hormone is actually incredibly selfish from the bacterial perspective, because all they're doing is promoting the plant to grow more so that they have more energy or more access to food sources in the future. So this 380 million year old relationship is well tuned, and it's well established, and it's worked out all the kinks. Next slide. So if we have this and it's out there and we know and understand about it, why don't we harness this ourselves for improved crop yield? So the microbiome offers an integrated approach to maximizing biomass. And in this instance, if you think about it more as the whole is more valuable than the sum of the parts. So in science, we typically, and we almost have to, try to break things down into individual components, isolate them, scrutinize them, interrogate them, and hopefully learn more about the system or the organism as a whole. But what we found, especially in the last 20, 25 years, is the systematic approach, or the systems approach to biology, in that there is definitely value in breaking down a whole into its parts and understanding the parts individually, but it turns out it doesn't really give us the whole picture, and we're not typically able to recreate these things when we try to tinker at them. If you approach things from a systems perspective, where you're looking at a holistic view and the entire relationship and all the moving parts in there, we tend to have a better understanding of how they work. We also tend to, if we can, be in control and have a better outcome for our own purposes. So if the Rhizosphere is the head office for the nutrient production and uptake, why apply exogenous chemicals equally across the bulk soil? So standard or traditional farming or ag practices would see the application of fertilizer to the soil. And although there's value in that, and we'll probably never get away from that completely, if you think about it from a pure economic perspective, you're putting down a lot of chemical that what we just understood about the plants and the plant root system might not ever be available to the plant. It can be leached off, it can be inactivated, it can be denitrified. So it typically falls into that waste category, and that you're not going to be able to capitalize off of what you put down there anyhow. Ideally, we'd like to locate these resources as close to where they are most utilized. In this case, it would be the root system. Next slide. So indeed, that's what happened with advanced ag in their ACF mixture here. They sought, or they endeavored to harness this 380-year-old million system for improved crop yield. And basically what they have is a cocktail known as the plant growth promoting bacteria. They've been introduced already a number of times this morning, so I'm not going to speak too much about them, but what you have is several different species, or 5 different species, that have been isolated from soils, that have particular functions that we understand are needed by the plant. We understand how they work, and we're able to create an environment for these bacteria such that they can act and impart these systems to the plant health. Next slide. So specifically, when we look at this, we have the Rhodopseudomonas and the Bacillus licheniformis, which actually enhance soil bacterial activity. So these are the guys that are going in there. They're going to be degrading and promoting, or degrading and then allowing for the promotion of growth or additional growth of bacteria through the production of broken down materials from the plant. This in turn allows these other guys to do their job. And in the context of what we care about, their job is to solubilize phosphates, improve nitrates concentrations, convert ammonia or other forms of ammonia to nitrate, as well as cytophore production, which is really just the availability or making ions, metal ions, available to the plants. So that's something that Bill spoke about a little bit when he's talking about arsenic earlier. Next slide. So if we break this down, you can see that there's a number of functions that each one of these performs on their own as a standalone, some of them more than others, but you're going to get in this cocktail is the ability of these guys to sort of feed off of each other. And I think the most important example that I can draw here would be when we're talking about nitrogen, or when we're talking about nitrogen fixation and nitrogen conversion versus phosphorus solubilization. So the interesting thing is when you want phosphorus soluble, ideally it wants to be in a certain pH range. So when you have high acidic or high alkaline soils, you don't tend to have your phosphorus be soluble, and it gets locked down or it gets leached away and gets taken. The plant can't absorb it, it can't use it. But when you have bacteria in there that are dropping off acidic ions, so ammonia, converting ammonia, you have the increase or the decrease in the pH, you have the increase in the alkalinity, which will allow certain soils to get into that pH range where there's more phosphorus available. So that's just a simple example of how you're actually using these guys off of each other, how the interplay between them, or the interspecies mutualism, allows this cocktail to create an ideal scenario for your roots. Next slide. If we look at real world evidence here, I think we're looking at soybean roots. When you look at the plants that are taken from a treated field or a treated plot versus an untreated, you can see that there's a significant increase in the root mass around at the bottom of on the left side in the treated one. Although we can't say that this necessarily translates to yield from this picture, it's okay, I guess, to assume that what you're getting out of that root system on the left is going to be significantly more than what you're getting out of a more underdeveloped system on the right. Next slide. Same thing here at the root systems. We're looking at a corn root system. Again, we don't have good metrics, so I couldn't say exactly what the biomass is, but even from a simple visual perspective, we can see that the example on the left, which would be the corn from a treated plot or a treated field, looks like it has much longer roots as well as many more shoots or root shoots. So what we would expect from that corn is that it would be more successful at taking from the ground what it needs and receiving what it needs in that transport system, hopefully converting it to above ground foliage and ultimately yield. Next slide. And finally, this would be on barley, and this actually is a yield figure. So we can see that in untreated control, that would be the black bars, yields a certain particular amounts of bushels per acre. However, when we treat it with the ACF, whether it be in furrow or foliar or in conjunction with something else, we're able to increase that base yield significantly so that you have a larger take off the field that is treated. Next slide. So in summary, what I hope you to take away from this is that the plant microbiome relationship is an ancient relationship through which plants and bacteria maximize their life potential. Agricultural producers can harness this relationship through soil inoculants, also known as plant growth promoting bacteria, an example of which is the ACF that we spoke about throughout the day today, really. And the use of this plant growth bacteria increases the concentration of usable nutrients in a spatially relevant fashion. So we're not necessarily putting nutrients equally everywhere across a field. We're trying to locate them specifically to be in that adjacent root region known as the rhizosphere, where they're most easily accessed and most useful to the plant. The increased nutrient availability can result in increased root bulk, increased foliage, and together we would expect that those things produce an increased yield to the agriculture producer. So the final thing I want to say is that the ACF is really a natural product. What we're trying to do is recreate this holistic approach or this systems biology approach. And you have this natural product that leads to healthier plants. And the healthier plants, in turn, hopefully will lead to healthier humans who consume them. And that's the end of my speech.